Hello everyone. Good morning. A very good morning to all of you. Am I visible, audible? Am I live? Can you see me? Can you hear me? Someone from the audience give me a thumbs up in the chat box. Let me confirm if I'm clearly visible, audible. I will start the class ahead. Just a second. Give me a second to confirm. Just give me a second. I hope it is working. Yes. I hope it is working. Just a second. Okay. So I welcome you all for today's session. A very good morning to all of you. I am Dr. Priyanka Sachdev here, and today I am here to continue my series of virology. So in virology, we have already covered general virology. We have already started the DNA viruses. We know that there are 6 DNA viruses. So the mnemonic is in front of you. We already know that is HH8PPP. These are the 6 DNA viruses in front of you. Till now, till yesterday's lecture, the first DNA virus which we have covered yesterday was pox virus. After that, I moved to adenovirus. After that, I moved to parvovirus. And after that, I have completed papovavirus. So those who are old students, they already know that we have already completed 4 of the 6 DNA viruses. Now, Hepadna and herpes DNA virus is remaining. After that, I have to start RNA viruses today. So let me start with herpes virus. So first I will teach you there are 8 type of herpes virus. All 8 are in your syllabus and it is very IMP topic. Many MCQs are asked from these herpes virus. You yourself will see. I will launch all the previous year questions in the polls. So I will teach you all 8 herpes virus one by one. I will teach you the classification and then Hepadna virus I will take with the RNA viruses. In Hepadna, basically hepatitis B virus is there. Hepatitis B virus. But hepatitis C, A, E, other viruses are in RNA viruses. So I will teach you all hepatitis together. So basically after herpes, I am going to start RNA viruses. So that is the plan today. That is the plan today. Shall we go ahead? I think we should go ahead. So you can see these are our headings under which we are describing all the viruses. I guess the list is ready with you. Your notebook and pen is ready with you. So till now in the pox virus, we have completed 3 viruses in the pox virus. That is variola, vaccinia and molluscum contagiosum. These 3 are the pox viruses. This table should be ready for you, with you for the pox viruses. After that, we have already completed adenovirus. Adenovirus is only one virus. There are 52 types, but we have not covered separately. Overall adenovirus I have taught you. After that, parvovirus is only one parvovirus B19. We have completed that also. I guess the list is ready with you. After that, papova, there are 3 viruses. Papova. The most important in papova is papilloma virus. That is human papilloma virus. We have completed that also yesterday. I guess till now this list is ready with you. Those who are the new students, they don't know about the list. I request them to watch the recordings of the previous lecture to complete this list. Now I am asking you to continue this list. So let me start now. Herpes virus. Herpes virus. So I will teach you 8 type of herpes. Herpes 1, 2, 3, 4, 5, 6, 7, 8. So I request you to divide the list into 8 more columns ahead. Or take a new page and take 8 columns in that so that we can complete all the herpes virus together. Give me a thumbs up so that all the DNA viruses till now what I have taught you will be on one page or in a tabulated paper, right? In a tabulated manner. So the thing is that when you will start revising DNA viruses at the end, you can revise entire DNA viruses in just half hour, right? So let me start herpes virus now. Herpes virus. So as we all know, the first heading is the morphology, right? In morphology, we always say about 5 things. What is the shape of virus? What is the genetic material of the virus? Whether the virus is enveloped or non-enveloped? What is the symmetry of the virus? Where does the virus replicate in the host cell? There is no peculiar shape. So make a cross, the peculiar shape, there is no shape. There is no peculiar name of the shape. Like pox virus was brick shape. These all were brick shape, right? You already know. ये तीनों pox virus हैं. यहाँ पे shape was brick shape. Adenovirus का specific shape था, if you remember, space vehicle. It was space vehicle. There was no specific shape here, no specific shape here, and there is no specific shape here. So these things I am telling you in general for all 8 type of herpes virus. So there is no particular shape. As we all know that herpes virus is a DNA virus, it is a double-stranded DNA virus. Only parvovirus is a DNA virus which is single-stranded DNA virus. Rest all DNA viruses are double-stranded. So herpes, all herpes are double-stranded DNA viruses. About enveloped and non-enveloped, you already know the mnemonic. Now I shouldn't tell you. HHAPPP. The third P is the pox, right? Among these, you already know, I guess everyone knows, these three are non-enveloped. These three are non-enveloped, and the remaining three, these three are enveloped. Now you yourself see where herpes is coming. Herpes and hepadna, both are enveloped. So of course it is an enveloped virus. About the symmetry, you already know HHA, PPP, the last pox virus is having complex symmetry, right? The remaining five have icosahedral. So herpes, I am interested in herpes. Herpes is having icosahedral symmetry. About replication, again you know, only pox virus, which is the largest virus in DNA, replicates in cytoplasm of the host cell. Remaining all five replicates in nucleus. So it replicates in nucleus. I am interested in herpes. So I hope everyone got it. Everyone got it. Give me a thumbs up. So that is about the morphology. After morphology, I will show you the diagram of the herpes virus. This is a diagram of herpes virus. I hope everyone can see it. Can you see? It is a double-stranded DNA. It is double-stranded DNA as a nucleic acid at the center, right? It is surrounded by a capsid. Can you see? This is capsid. This one is the capsid. It is surrounded by a icosahedral capsid. This is icosahedral shaped capsid. Now you all can appreciate there is envelope. This one is the envelope. Since it is an enveloped virus, no? It is an enveloped virus, we know. So we can see the envelope. We can see the envelope also. So in the capsid, I'm interested in capsid. In the capsid, how many capsomers are present? Tell me the number of capsomers in the capsid. Have you remembered the capsid? Yesterday I taught you the capsid of adenovirus also. Adenovirus was space vehicle shape. If you remember, I told you 252 capsomers are there, out of which 240 are present in the icosahedral symmetry, and the remaining 12 are forming the rod-like projections which give the shape like a space vehicle. If you are the old student, you may be knowing it. Now I am saying you all herpes, 8 type of herpes. There are 162 capsomers, 162. So in some viruses, this number is important. I'm not telling you this number in all the viruses. You will say, ma'am, you have not told us the number of the capsomers here, here, here. So it is not important there. Wherever it is important, I'm telling you. So till in DNA viruses, only two places you have to learn the number of capsomer in the capsid. One is an adenovirus, that is 252, giving a space vehicle shape, and one is an herpes virus, that is 162. You can see here the number of the capsomers in capsid is 162. You have to learn this number. There are 162 capsomers, right, which forms the capsid, right? Now the capsid is surrounded by an envelope. We can see the envelope. Now you can see a space. You can see a clear space between the capsid and the envelope. What is this space? The space between the capsid and envelope is filled with a pink color material. Can you see that pink color material? That pink color material is known as tegument. It is an amorphous material. That pink color material is known as tegument. Between the envelope and between the capsid, there is an amorphous material that is known as tegument. Right? That is known as tegument. So that's it. And apart from it, there are papillomers. You can see the spike-like projections on the envelope. So this is the diagram. If this diagram is coming in your exam in the form of a IBQ, you should be able to identify it. So there is a double-stranded DNA at the center. It is a double-stranded DNA. It is surrounded by a capsid having 162 capsomer. Learn the number. And it is surrounded by an envelope which is made up of lipid. This is the envelope. And there are spike-like projections on the envelope which are made up of glycoprotein. These are glycoprotein spike-like projections. The space between the capsid and the envelope is filled with the amorphous material known as tegument. That's it. I want thumbs up from everyone. So if this diagram is coming in your exam, you should be able to identify, ma'am, it is a diagram of herpes virus. All 8 herpes virus have same diagram. If space vehicle is coming in your exam, you may be remembering it is adenovirus. If brick shape is coming in your exam, you may be remembering it is pox virus. So basically, you should know the diagram of all. You should know the diagram of all viruses for your image-based questions. Give me a thumbs up. Give me a thumbs up. After diagram, I would move like to one more important body. I have taught you inclusion bodies. In general virology, those who are the old student, they may be knowing what are inclusion bodies. Inclusion bodies are of 3 types. They are seen inside the host cell. They can be present in the cytoplasm of the host cell. They may be present in the nucleus of the host cell, and they may be present at the both. Right? Currently, I am teaching you herpes virus. So herpes virus में inclusion bodies होती हैं क्या? Does herpes virus have inclusion body? Yes. The answer is yes. Now you tell me where does it come? So herpes virus inclusion bodies are cytoplasmic or nuclear or both. I am giving you 3 options. I have taught you the examples. Now you may be knowing examples of intracytoplasmic inclusion bodies. You may be knowing the examples of intranuclear and you may be knowing the examples of both. I have taught you this thing very clearly in general virology. Yes or no? I have taught you. So you tell me, where is herpes virus coming? In which example? I'm asking to you. Yes, S49413. You are absolutely right. It is nuclear. Let me revise to other students. Okay. So intracytoplasmic, these are the examples I have given you. In intracytoplasmic, it is rabies virus, in which Negri bodies are there. Vaccinia, in which Guinea, Variola, in which Pasteur, Foulpox, in which Bollinger, and Molluscum contagiosum, in which Henderson-Peterson. So these all are pox viruses, the various types of pox viruses along with rabies virus. Rabies virus. So rabies virus is an RNA virus and pox viruses are the DNA viruses. So the rabies and pox have cytoplasmic. In short, you can say. In short, you can say cytoplasmic inclusion bodies are present only in two viruses. Number one, rabies virus, which is an RNA virus. I will teach you rabies in RNA viruses and the various types of pox virus and learn the name of their inclusion body. So I have told you this very clearly. Coming on nuclear. In nuclear, I told you there are two types of nuclear bodies. What are the two types? Quadrid type A and quadrid type B. What do you mean by that? That is the difference. Now, of course, both inclusion bodies are present inside the nucleus of the host cell. That's why they are nuclear, intranuclear. But quadrid type A is granular. Quadrid type A is granular in appearance and quadrid type B is circumscribed in appearance. So appearance is different. Now see, what is our virus today? Today our virus is what I am teaching you right now. I am teaching you herpes virus. So herpes simplex virus is having quadrid type A. Yes, S4, absolutely right. Herpes simplex virus is having quadrid type A. The name of the body is known as Lipschul's body. Variola zoster is a type of herpes only. So I will teach you 8 type of herpes. Herpes 1, 2 and 3. They all have inclusion bodies. That is quadrid type A. And yellow fever also have it, which is known as Story's body. Right. Quadrid type B is present only in P and A. So polio and adeno. Give me a thumbs up. And the both are present only in two viruses, measles and CMV. So that is the thing. CMV is also a type of herpes virus. Let me teach you the class. So you know the inclusion bodies, right? Let me teach you the classification of herpes virus. In herpes family, herpes is a family. Herpes is a family in which there are 3 subfamilies. And under the 3 subfamilies are alpha, beta and gamma. And under the 3 subfamilies, total 8 members are there. Total 8 members are there, right? What are the 8 members? Herpes virus type 1, type 2, type 3, type 4, 5, 6, 7, 8. Total 8 members are there. Some of them belong to alpha subfamily, some of them belong to beta, some of them belong to gamma. So these are the 3 subfamilies under which total 8 members. So there are 8 members in a family. 8 members in a family, type 1, 2, 3, 4, 5, 6, 7, 8. You have to learn their other names. You have to learn their other names. Type 1 is known as HSV1, herpes simplex virus 1. Type 2 is known as HSV2, herpes simplex virus 2. Very good. Type 3 is known as VZ, varicella zoster. It is not variola, it is varicella. This is the virus which will cause chickenpox. Right? Smallpox kaun karata hai? Who is causing smallpox? I have taught you smallpox is caused by one of the pox virus, variola. Chickenpox is a misnomer. इसमें suffix में pox है, but chickenpox is not caused by pox virus. Chickenpox is caused by one of the herpes virus. It is herpes 3, which is causing chickenpox. The name of the virus is varicella zoster virus, VZ virus. Type 4 is Epstein-Barr virus, EBV virus. Type 5 is CMV, cytomegalovirus. Type 6 is human B cell lymphotropic virus, HBLV, human B cell lymphotropic virus. Type 7 is RK virus or Kaposi virus. I have to confirm 7 ही है क्या? It is RK virus. And type A is Redinovirus. You have to learn their names. Right? So इसमें से अल्फा में कौन कौन आएगा? 1, 2, 3 are coming in alpha. बीटा में कौन कौन आएगा? 5, 6, 7. 1, 2, 3, 5, 6, 7. और गामा में कौन कौन आएगा? 4 and 8. So these are the 3 subfamilies you should learn. So you should always know the classification. Everyone give me a thumbs up. Everyone means everyone give me a thumbs up. Just a second, we will do some questions based on the classification. You got the classification? You want me to say it again? I guess I should say it once. Okay. So 1, 2, 3, 4, 5, 6, 7, 8. The 8 types of herpes virus. Who will tell me the other names? Would you like to try? Pankaj, Lavanya, S494, anyone? Pankaj. Okay, Satish, you would like to help. Okay, tell me the name of type 1. It is herpes simplex virus 1, HSV 1. What is type 2? It is HSV 2, herpes simplex virus 2. What is type 3? It is VZ, Varicella Zoster virus. What is type 4? It is Epstein-Barr virus, EBV. What is type 5? It is CMV, cytomegalovirus. What is type 6? Type 6 is human B cell lymphotropic virus, HBLV. Type 7 is RK virus. And type 8, I have to see what is the name of type 8. I just forgot it. So there are 8 type of viruses and you have to learn their names. Yes, you all are right. Soumya, Pankaj. Very good, very good. Right. So you have to learn their names according to their numbers and you have to divide them in their subfamilies. There is one question. I guess only one question is there. Which of the following belongs to Herpes viridae family? Which of the following virus belong to Herpes viridae family? Variola virus, adenovirus, HPV virus, ki RK virus. The 4 options are in front of you. The 4 options are in front of you. Who will tell me the correct answer? Who will tell me the correct answer? I guess many of you already started answering answer. Yes. So, Pratyaksha, Prishinika, very good. What is the correct answer here? I want everyone to participate. What is the correct answer? Yes, Soumya and Kian. Yes, Pankaj. Correct answer here is D. So, RK virus. Which virus? It is type 7 virus. Type 7. Okay, tell me the families of other. Ye तीनों कौन सी family के हैं? Ye तीनों, these are not herpes family. I know. So, which family they belong to? They belong to which family? They belong to, okay, variola belong to which family? Variola belong to pox viridae. Variola belong to pox family. Adeno belongs to adeno family. Adenovirus is a member of adeno family. HPV belongs to which family? Tell me the name of HPV. HPV कौन सा family को belong करता है? HPV belongs to which family? It is Papova. Papova का पा है ये. There are 3 viruses ना. So papilloma, human papilloma virus. So you should know the families of the others and RK belongs to herpes family. See, I have written the 4 names in front of you. Now I can change the question now accordingly. Instead of herpes virus, if I am changing the question, which of the following belongs to pox viridae? Pox viridae, your answer will become A. Which of the following belongs to Papova viridae? Papova, your answer will become C. So likewise, we can change the question. We can change the answer. Everyone give me a thumbs up if you got it. Everyone. So you can see the correct answer here is RK virus, right? So, okay, here also from one other book, we can see the classification. It is HSV1 type 1, HSV2 type 2 and VZ virus. VZ is type 3, right? And Epstein-Barr virus is type 4. Cytomegalo is type 5. Human B lymphotropic virus is type 6. Type 7, okay, we got what is type 7 is RK virus. Type 8 का other name भी याद करो, Kaposi. Kaposi virus is the type 8. Type 8 के 2 names are given. What are the 2 names given to type 8? Type 8 को Redino भी याद करो और type 8 को Kaposi भी याद करो. Learn both names. So these are the names given to them. Everyone give me a thumbs up. Everyone means everyone. Now, I will teach you all 8. Don't worry. All 8. Is it blood? Pankaj is saying and it is looking blood. Is it so? No, from my side, upload is absolutely clear, Pankaj, I guess. What about others? Am I clear or am I blurred? Write down in the chat box. Okay, clear from others. I guess the problem is in your download, Pankaj. So, okay, let me move ahead. So I will teach you all 8 one by one. So I will start with type 1, HSV1 and HSV2. I will teach you together. HSV1 and HSV2 I will teach you together. These two I will teach you one together. Then I will teach you these separately. So let me start HSV1 and HSV2 together. HSV. So human herpes simplex virus, type 1 and type 2, right? So let me start with this. So there are 2 type of HSV, 1 and 2. HSV1 is type 1 herpes and HSV2 is type 2 herpes, right? So these are 2 different viruses. These are 2 distinct members of the family, herpes family. 2 distinct members are there. So, okay. What is the difference between them? Basic difference between them, HSV type 1 spread from one person to another by respiratory route. By droplets. HSV2 spread from one person to another by sexual route. Right? So that is the first difference. The route is different. Right? I'm coming on that also. I'm coming on that. So both will cause rash. Okay. HSV type 1 HSV type 2. HSV type 1 transmitted from one person to another person by droplet infection mostly, and HSV 2 transmitted from one person to another by sexual route, right? Okay, so there are differences between HSV type 1 and 2. I will come on this table. Let me explain you the pathogenesis of both, right? Both will cause lesions. Okay, let me draw a diagram. Both will cause the rash. Both will cause the rash. Now there is a general rule where they cause rash, which part of the body they cause rash. So let's draw a line at the waist, at the point of the umbilicus. So above waist, the rash is caused by HSV1, and below waist, the rash is caused by HSV2. This is a general rule overall. So let me show you a few diagrams. I will come on these tables again. The reservoir of both HSV1 and 2 is human mucosa and ganglia. That is the reservoir. Transmission, direct contact for HSV1, sexual contact for HSV2. We are taking two together. Let me teach you the pathogenesis. The most important, the pathogenesis. The most important thing is the pathogenesis. Okay. Listen, the primary infection of HSV1 and 2 both occurs either through skin or oral mucosa or eyes. So either it is genital infection or through droplet infection, the virus is coming in the human body. After coming in the human body, after entering in the human body, it will go in blood. From the blood, it will go to the skin and it will form a vesicle in the skin. I will show you the diagram, just a second. So these are the vesicles. Can you see? These are the small vesicles. In HSV1, you can see around the mouth. You can see small vesicles. In HSV1, you can see small vesicles. These are on genitals. So this is HSV2. So this one is HSV2 because it is on the genital, below waist. Here the above waist. So vesicle is in both. The rash or vesicle, the rash is in the form of vesicle formation. So vesicle formation occurs in both. But if it is above waist, it is HSV1. If it is below waist, it is HSV2. So here it is in mouth, so of course it is HSV1. Here you can see again it is face. So again it is HSV1. But here it is genital area. This is the penis of a male. This is the vulva of a female. So it is HSV2, right? Now whether it is HSV1 or HSV2, the zoom version is like this. These are the vesicles. These are the vesicles. Give me a thumbs up. Can you see? These are the vesicles. So the location is different, but morphology is same. It can occur at the fingertip also. Fingertip pe bhi ho sakte hain. So it can occur around the mouth, around the lips also. The vesicles are there. The vesicles are there. Can you appreciate? Can you appreciate it? Give me a thumbs up. So some of them are caused by HSV1 and some of them are caused by HSV2, depending that whether it is above waist or below waist. So first, whether HSV1 or HSV2, let me write both here. HSV1, HSV2, both of them enters in human body. The route is different. HSV1 will enter by respiratory route, droplet route or direct route. HSV2 will come in the human body by sexual route. But once they come in human body, after going in the blood, they will go in the skin and form the vesicle. This is known as primary infection. This is known as primary infection. In some days, the vesicles will be cleared and scarring will be there. Like in chickenpox, we have scarring in the end. No? So vesicle will mature, it will rupture, and in the end, we have scarring. The human have scarring, the patients have scarring. But the virus is not dead. The virus have a special feature. After primary infection, the virus go to the sensory nerves and remain in the ganglion of the sensory nerves for years and years. It remains latent there. It remains dormant there. It remains there for many years. So which ganglia? If it is HSV1, it remains in trigeminal ganglia. If it is HSV2, it remains in sacral ganglia. So it remains in sensory nerve ganglia in the human body, hidden, chupke baitha hua hai, chupke. Now when the immunity drops, it again reactivated. And at that time, again it produces symptoms like common cold, fever, pneumonia, stress, and again vesicles will be formed. This is known as latent infection. Give me a thumbs up. You got my point? Yes or no? So listen, let me take a whiteboard. Just suppose this is the skin of the person. Initially the virus is entering the first time, HSV1 or HSV2. After coming in the blood, it is coming on the skin and forming primary vesicle. So these vesicles are primary vesicles. This is a primary vesicle, it is formed. From here, the virus is going. Now it is resolved, scab is formed. Now this is a nerve. This is a sensory nerve. Basically, this is a sensory nerve. Sensory nerves go to the brain. Now this is the brain. It carries signal to the brain. This is a brain, right? And in between, these are the ganglions of the sensory nerve. These are the ganglions on the sensory nerves. We know what are ganglions. So the virus is traveling retrograde. The word here to be taken is retrograde. Ulta ja raha hai virus. It is going retrograde towards the brain, and just suppose this is the virus. This is the virus. So from the vesicle, it is going retrograde in the nerves and get hidden in these ganglions. If it is HSV1, it is hidden in trigeminal ganglion. If it is HSV2, it is hidden in sacral ganglion. दोनों पे MCQ है. HSV1 hidden in trigeminal, HSV2 hidden in sacral. Now, it will remain there for years and years. When the immunity drops, it again is reactivated. After becoming reactivated, it again come the antigrade route. पहले retrograde से अंदर गया, अब antigrade से वापस ऊपर आके it will again form vesicles. Again it will form vesicles, and now this is secondary infection. This is latent infection. So latency is the hallmark of these viruses. These viruses have latency. You know the meaning of latency or dormancy. They have a feature, special feature, which is not present in other viruses. Latency or dormancy. Give me a thumbs up. Everyone, not only in HSV1, HSV2, all 8 herpes family virus have a special feature of dormancy. Give me a thumbs up. So here also you can see the same thing. So entry of virus by skin or by mucous membrane. So virus multiplication in the blood. After multiplication in the blood, it is coming in the cells of the skin. It is forming vesicles. It is causing ulcers. So this is primary infection. This is primary infection. From here, virus is going to the sensory nerves. Retrograde. This is retrograde, right? From there, it is going to the ganglion. Which ganglion? Either trigeminal or sacral, right? If it is HSV1, it is trigeminal. HSV2, it is sacral. Remain dormant there, remain latent there for years. When immunity drops, it causes reactivation. Immunity drops either in cold or fever or surgery due to any reason, due to stress. Immunity drops, then there is reactivation, and again it will form vesicles. So at that time, the vesicles are secondary vesicles. The same is shown in the diagram. The same is shown in the diagram also. You can see the same is shown in this diagram also. Okay, so these are the vesicles produced. You can see first it is going, remain hidden here in the trigeminal ganglion, and again it is coming back and forming vesicles. So same thing. So disease is basically incubation period is 6 to 8 days. Okay, now primary HSV infection is more severe and associated with more symptoms and complication as compared to latent. Both are same. दोनों में vesicles बनेगा, दोनों में rash आएगा, but primary is more severe as compared to latent. That you should know, right? So at what site vesicles can be formed? Vesicle can be formed around the oral facial and oral. That can be done. So it can be formed on the face, it can be formed around the mouth. So there are various types of herpes. Herpes labialis is forming around the mouth, right? And gingival mastitis or pharyngitis can occur. So in the pharynx also, inside the mouth also, outside the mouth also, at the all the places oral vesicles can be formed. Number 1. So this is by HSV1. I guess it is by HSV1. Second is genital. It is formed at the penis in the male and cervix and urethra in the female. So it is caused by HSV2. HSV2. These are painful. These are bilateral. I have shown you the diagram also, I guess. This one. So you can see males may it is penis and females may it is urethra, cervix, vagina, right? So that is the thing. Just a second, right? Just a second, right? Now one more thing, it can be formed around the finger. It is known as herpetic whitlow. Around the fingers, this diagram. It is herpetic whitlow. Herpetic whitlow. Around the fingers, it is known as herpetic whitlow. Give me a thumbs up. Around the. Now in wrestlers, you know what is wrestlers? Wrestlers have a punch. Usually they have a punch on the face. So they have a punch around the lips. So this area is very commonly infected in wrestlers. So they can have herpes in this area. It is known as herpetic glandiatorum. Let me show you this one. Herpetic glandiatorum. This is usually seen in wrestlers because of the punch. In this area over the lips, this area commonly infected. Give me a thumbs up. Everyone give me a thumbs up. So you should know the various names only. So the thing is same. It can be around the eye also. It can be in the brain also, causing encephalitis, meningitis. It can be in visceral other organs also. The vesicles can be formed in the esophagus. It can be formed in the lungs. It can be formed in the trachea. It can be disseminated throughout the body. It can be in internal organs. So vesicle can form anywhere in the body. It is not only in the skin. It can be anywhere in the body. So the various types of vesicles. So what is the general rule? Now some of these vesicles are formed by HSV1. Some of these vesicles are formed by HSV2. So this is the general rule. Vesicles above waist are caused by HSV1 and below waist are caused by HSV2. I have drawn the diagram also. I am drawing it again. So this is a person at the level of umbilicus divided. So vesicles formed above this region, it is caused by HSV1, and below this region, it is caused by HSV2. I want thumbs up from everyone. Come on. So this is a rule of thumb. You can learn like this. So you have already seen the images. Let me proceed now. So we have done the infection, right? The same thing is shown to you. So whether it is HSV1 or whether it is HSV2, HSV1 causes usually oral lesions in primary infection. Oral lesions or lesions above the waist, right? HSV2 usually causes genital lesions, genital vesicles in primary infection, that is below waist. HSV1 remain latent in trigeminal ganglion for years. HSV2 remain latent in sacral ganglion for years. Yes or no? Yes or no? So HSV3 I am going to teach you next. HSV3 नहीं मतलब herpes type 3. Herpes total 8 types का है ना. So herpes 1, herpes 2 is HSV1, HSV2. Herpes 3 क्या है? VZ, varicella zoster. That is also causing latency. So in primary vesicle, it is chickenpox. उसमें VZ में, which I am going to teach you next, the third one. So this is type 1, this is type 2, this is type 3. Out of the 8 type, third one मैं अभी लूंगी. That is varicella zoster virus, right? So इसमें actually दो viruses हैं, varicella and zoster. Varicella cause the primary infection and zoster cause the secondary infection. So two viruses are there. So varicella cause chickenpox and zoster cause shingles. Zoster, both are vesicles, both are rash only. Actually, hair rashy. So the latency is there, but the ganglion can be any sensory ganglion. There is no specific ganglion. So this is the comparison of type 1, 2, 3. Let me proceed now. What is the lab diagnosis? Let me finish type 1 and 2. 3 pe main aati hu. I will teach you the 3 ka details. Let me finish type 1 and 2. Type 1 and 2, you got it? So this is HSV type 1 and 2. Some of them are 1, some of them are 2. Right? We got it. Everyone give me a thumbs up. Lab diagnosis mein, what is your specimen? Your specimen is the vesicle fluid. Basically, your specimen is the vesicle fluid. Now, the most important, can you do a light microscopy? Yes, you can do a light microscopy. In light microscopy, there is a there is a typical smear which is known as T-Zank smear. What is this T-Zank smear? You take a vesicle. From the base of the vesicle, you take scrapings. So can you see any vesicle? Can you see this vesicle, or this vesicle, or this vesicle? You just rupture it with a sterile needle and take the base. Take the base out. Take the scrapping of the base. Scrapping of the base on a slide. You take a slide, rupture the vesicle. You take the base of the vesicle on the and make a smear of the base of the vesicle. You are making a smear from the base of the vesicle. What you are doing? You are taking the base of the vesicle. You are making a smear and you are staining it with toluidine blue. You are staining the smear. So what basically you are doing? You are taking a slide. You are rupturing the vesicle. From the base of the vesicle, you are taking a fluid and making a smear. And you are doing the staining by toluidine blue. This smear is known as T-Zank smear. T-Zank. First understand what it is. It is Zank. Yes. So T-Zank or T silent. So Zank is known as, it is T-Zank smear, right? Or Zank smear. So what you will find in the vesicle, from the base of the vesicle, you are taking the scrapings. So you are taking the cells. You are taking the cells of the host. So vesicle ke base pe kya hai? Okay, so this is the base of the vesicle. This is a vesicle. In the base of the vesicle, these are the host cells inside which virus is already entered. The virus is already entered. Virus is replicating inside. Now you are taking these cells on a slide. So this is one of the host cell. You are taking this cell out. So this is one of the host cell inside which virus is present. We cannot see virus on light microscopy. We all know. So we will see the other features. We will see the inclusion bodies inside the Zank smear. Number 1. We will see the ballooning and syncytia. Not direct virus. These are the indirect indicators of virus on a Zank smear. Give me a thumbs up. So let me show you. So can you see the cell become multicellular? This is a big cell having multiple nucleus. Multicellular giant cell is there. You can see this is multicellular giant cell having multiple nucleus, right? You can see this is Caudery type A inclusion bodies. Inside the nucleus there is an inclusion body. So these are the indirect indicators that virus is present on light microscopy. But on electron microscopy, you can directly see the virus. You can always do the virus cultivation on a tissue culture, cell culture. You can do the PCR, you can do the serology. You know the ways how to diagnose the virus, right? Let me teach you the treatment of HSV1 and 2. HSV1, HSV2, that is type 1 and type 2, right? So acyclovir is the drug of choice. Acyclovir is the drug of choice. Otherwise, you can also use valacyclovir and famiciclovir. But acyclovir, if not available, you can use these also, and that is the drugs. That is the main drugs we use here. So out of the 8, 2 are done. 1 and 2. Give me a thumbs up. So can you tell me their pathogenesis? Can you tell me the name of the diseases they are causing? Can you tell me their lab diagnosis? Can you tell me their treatment? Everyone give me a thumbs up. Jitne audience utni thumbs up. You got it? You got it? Yes or no? Yes or no? Say. So their route of transmission is different. HSV1 is transmitted by fomites, respiratory route, and HSV2 is transmitted by sexual route. Give me a thumbs up. Give me a thumbs up. We would like to do some polls on type 1 and type 2. Then we will move on the third type, type 3, that is Varicella zoster, VZ. Type 3 पे आएंगे, 4 पे आएंगे, 5 पे आएंगे. We will study all one by one. But before that, some polls on 1 and 2. That is HSV1 and HSV2. That is the name of 1 and 2. Right? So the first question is in front of you. Lipschule's body. That is the name of inclusion bodies. I guess the name of inclusion bodies are seen in which virus? So Lipschule bodies are seen in Hodgkin's disease or viral hepatitis or herpes virus or influenza. So I think the question is super easy for all of you. It's a super duper easy question and everyone is right. So Pratyaksha, Bodko, Soumya, you all are right. The correct answer, of course, it is herpes. Not only herpes type 1, type 2, all 8 herpes. All 8 type of herpes have inclusion bodies, which are Lipschule's bodies. These are the type of nuclear, intranuclear inclusion bodies, and these are Cowdry type A. Cowdry type A means these are granular. We have already seen it. So many questions can be created, not only this, based on the inclusion bodies. Yes, the correct answer here is C. It is Cowdry type A, granular nuclear bodies, intranuclear. The second question in front of you, herpes simplex virus. I am asking about HSV. I have not mentioned 1 or 2. So it can be anything. If I have not mentioned the number, it can be anything. You tell me the genetic material. Is it DNA or is it RNA? Is it single stranded? Is it double stranded? The 4 options are in front of you. So first decide it is a DNA virus or RNA virus. Or if it is a DNA virus, is it single stranded or double stranded? So you already started answering. Ayush, Pratyaksha, Rishinika, Bodko, you all are right. Very good. Very good. So Pankaj, Soumya, very good. What is the correct answer? What is the correct answer? Yes. So it is a double-stranded DNA virus. All DNA viruses, there are 6 DNA viruses, HHA-PPP. All of them are double-stranded except Parvo, which is single-stranded. So herpes is a DNA virus which is double-stranded. So answer is B and you all are right. Yes. So it is DNA virus. The next question. The next question is in front of you. Okay, just a second. I have not taught you this, but just give your guess, then I will tell you the correct answer. HSV affects which area of the brain? These are the various options in the brain, the various portions in the brain. So various portions in the brain, basal ganglion, temporal lobe, cerebellum or pons. So HSV affects which portion of the brain? I have not taught you this, but if you know this, you tell me. Otherwise, I will tell you the correct answer. No, Pratyaksha, Rishinika, it is not the correct answer. It is not the basal ganglia. I know it is a sensory ganglia. I have told you the name of the 2 sensory ganglions. It is trigeminal ganglion and sacral ganglion. It is not basal ganglion. It is not basal ganglion. No, it is not basal ganglion. It is the answer is temporal lobe. If the HSV is going in the brain, if HSV, I'm not talking about latency. Please understand the question first. I'm not asking about latency. कि ये जाके छुपता कहाँ है, मैं वो नहीं पूछ रही हूँ. मैं पूछ रही हूँ if it is going in brain, it will affect which portion of the brain. I have taught you HSV can cause vesicles technically in every part of the body. From head to toe, it can produce vesicles in any part of the body. So above waist, below waist. Above waist, if it is forming vesicles inside the brain, it will cause encephalitis and meningitis. I have taught you. So if it is causing encephalitis and meningitis inside the brain by producing vesicles in the brain, lesion in the brain, which portion of the brain? Which portion? So answer is temporal lobe. So it is written in my notes, but I forgot to tell you. So you please learn it. So what is the correct answer? You tell me. What is the correct answer? You tell me, please. So the correct answer here is temporal lobe. So most common acute viral encephalitis in most of the world is HSV encephalitis, and here temporal lobe most commonly affected, right? So virus is hidden in sacral ganglion for years, and during reactivation it will go to the brain and cause affection of the temporal lobe. Please learn it. Give me a thumbs up. Give me a thumbs up. Everyone give me a thumbs up. So herpes simplex virus causes all of the following except. Herpes simplex virus causes all of the following except. Does herpes simplex virus causes encephalitis, producing lesions in the brain? Does it causes pharyngitis, producing lesions in the tonsils? Does it causes infectious mononucleosis, or does it causes Whitlow, herpetic Whitlow? So which is not caused? You have to tell me which is not caused by herpes simplex virus. By herpes simplex virus. Who will tell me the correct answer? Who will tell me the correct answer? Yes, you all are right. The correct answer here is infectious mononucleosis. So infectious mononucleosis is caused by which virus? Tell me the name of that virus. Infectious mononucleosis कौन कराता है? Which virus? Can you tell me the name of that virus? Pankaj, Lavanya, Amalgame, Soumya. Can you tell me the name of that virus which causes infectious mononucleosis? Yes, I agree. Encephalitis, pharyngitis, and Whitlow. What is Whitlow? It is on the finger. I have shown you the diagram also. On the finger, if vesicles are formed, it is known as Whitlow. And in wrestlers, if it is caused by around the mouth, it is known as glanditorum. So it is caused by herpes simplex virus. Yes, you all are right. It is EBV. EBV, Epstein-Barr virus. That is type 4. Type 4. It is not type 1 and type 2. Type 4 is Epstein-Barr virus. So infectious mononucleosis is called not Ebola virus. It is Epstein-Barr virus. I will tell you when I will teach you type 4. Currently, I have taught you type 1, 2. अभी मैं 3 पढ़ाऊंगी. 3 के बाद when we will come on 4, I will teach you infectious mononucleosis there. So it is Epstein-Barr virus. Right. So that is the thing. Let me move ahead. The correct answer here is, you can see, infectious mononucleosis is caused by Epstein-Barr virus. And rest of the things given here are caused by herpes virus. Herpes virus causes all these. It can cause the herpes blisters, herpetic Whitlow, that is on the fingers, eczema herpeticum, right? Kaposi, varicella eruption, gingivostomatitis on the buccal mucosa, on the cornea of the eye, dendritic ulcer, in the brain encephalitis. These all are caused by herpes virus. Let me start the third virus now. Till now, out of the 8 viruses, I have to teach you 8 viruses today. 1, 2, 3, 4, 5, 6, 7, 8. The 8 type of herpes. The 8 type of herpes we have to study. Till now, we have completed type 1. The name of type 1 is HSV1. And we have completed type 2. The name is HSV2. Right? Both of them having same pathogenesis. Pathogenesis is same. That is the most important feature in the pathogenesis is the latency. But the route of entry is different in each of them. The lesions are same, but the location of lesion is different in each of them. One producing lesion above waist, one producing lesion below waist. Diagnosis is same, treatment is same. So we have done HSV1 and 2. Let me start the third herpes type 3. The name of the herpes type 3 is Varicella zoster. So starting the third herpes, Varicella zoster. I want thumbs up from everyone so that I can start Varicella zoster. First, give me a thumbs up. Everyone give me a thumbs up. Should I start Varicella zoster? I should. So Varicella zoster, it is said in some books, these are 2 different viruses. Varicella is a different virus, zoster is a different virus. So Varicella and zoster, they both combine together and form one virus. So okay, listen. So in this virus also, like herpes simplex virus, HSV1 and 2 latency is there. So when they enter the first time in human body, they cause primary vesicles. So primary vesicles, and they get hidden in the sensory ganglion. In the sensory ganglion, they are latent. They are latent for years. Then during the drop of the immunity, they become reactivated. And during reactivation, they produce secondary lesions. Right? So the primary lesions, the disease is known as chickenpox. It is known as chickenpox. You know, chickenpox. It is caused by varicella. It is caused by varicella. The primary infection is caused by varicella. And secondary is known as shingles. Shingles. This disease is known as shingles. So during reactivation, it is known as shingles, and it is caused by zoster. So it is said that. So varicella causes chickenpox, which is the primary infection, and zoster causes shingles, which is the secondary infection. And in between the two, the virus is hidden in the ganglion. There is no specific name of the ganglion in any sensory ganglion. There is no specific name like herpes, HSV1 remain latent in trigeminal. HSV2 remain latent in sacral. There is no specific name for this, but yeah, it can get hidden in any sensory ganglion. Give me a thumbs up. Everyone give me a thumbs up. Everyone means everyone. You got it? So that is the thing. So let me move ahead. The same thing is written in front of you. I have taught you. So chickenpox is a primary infection. And shingles, the other name of shingles is herpes zoster. That is caused by reactivation of the virus when the immunity falls. So it is a secondary infection, right? So what I am saying you, so there is a virus known as Varicella zoster virus, right? Varicella zoster virus. So when the virus enters the first time in human body, it causes chickenpox. Chickenpox. And chickenpox is caused by varicella, right? So it is a primary infection. So virus remain latent in human body for years in sensory ganglion, right? Now it becomes reactivated. It becomes reactivated. And during reactivation, it causes a disease known as shingles. Either shingles or herpes zoster. Herpes zoster, one and the same thing. The two names, herpes zoster or shingles. It is caused by zoster virus. And this is the secondary infection. In both of them, rash is there. In both of them, rash is there. Is there any difference in the rash? Is there any difference in the rash of the primary and secondary? Yes, there is a little bit difference. The little bit difference is that, let me draw a human body. Let me draw two human bodies. So first time if the virus is entering, it will cause chickenpox. You know, chickenpox rash occurs everywhere on human body except palms and soles. Except palms and soles, the rash can occur any part of the human body. Any part of the human body, sparing only palms and soles are spared. You can see the palms and soles are spared. Otherwise, rash can occur at any part of the body. Any part of the body. So you may be having, I was having chickenpox when I was a kid. So I can remember. So I was having rash everywhere on the body. So chickenpox is very common, right? Most of the children in India have it, chickenpox, right? That is the primary infection. That is caused by varicella virus, right? Now it got hidden. And during reactivation, the zoster component will be reactivated. Now it will produce secondary infection, right? The name of the disease is shingles. Shingles or zoster. Shingles or herpes zoster. So here, where is the rash? So let me draw the rash. Here the unique feature is there, rash is unilateral. It do not cross the midline. What do you mean by that? Rash do not cross the midline. Let me draw a midline of this person. So either the rash will be on the right side or the rash will be on the left side. Never both. You got my point? You got my point? Let me draw the rash. Just suppose rash is on the right side. So it is only limited on the right side. It will not cross the midline. So secondary never crosses the midline, never crosses the, or it will be on the opposite side. So it is not crossing the midline. So rash is unilateral, either right or left, never crosses the midline. But here in chickenpox, rash is on bilateral. It is both sides. Give me a thumbs up. I tried my best in the diagram. Everyone give me a thumbs up. Right? You got it. So these are the two diagrams, the same diagrams I will show you here also. So the property of the virus is that only one antigen difference is there in varicella and zoster. Okay, not important. So let me teach first varicella. बाद में zoster की बात करते हैं. So first let me teach you varicella. Then I will teach you zoster. So the two viruses, varicella, zoster, both are type 3 virus, type 3 herpes virus. Varicella causes chickenpox, right? It is herpes type 3 virus. Incubation period is 14 to 16 days, average 15 days. So whenever the virus enters in human body, after 15 days, the person have rash. The person have rash after 15 days. What is the source of infection? It is person to person contact. If your roommate is having chickenpox, तो भाग जाओ छोड़ के. You are going to have it because infectivity is very, very high. The infectivity is very, very high. Mode of transmission is air droplet, right? So whenever a person is having chickenpox, this person is having chickenpox. This person is having rash all over the body. He is a known case of chickenpox. So whenever he is having chickenpox, he is also having cough and coryza at the same time. Cough, coryza, sneezing. So by a respiratory route, it is spreading the virus to other person. So mode of transmission is respiratory route. So via respiratory route, other person is getting the infection via respiratory. So it is present in the aerosol. And rate of transmission, the infectivity is very, very high like COVID. So you had, so infectivity is nearly 90%. So if your roommate is having chickenpox, 90% chance is there that you will also have. If anyone at your home, any of your member, mother, father, children, anyone having chickenpox, 90% chance you will also have it. So infectivity is very, very high, right? So the mode of transmission is air droplet. Give me a thumbs up, respiratory. And source, it spread from person to person. Now there is a 2R5 rule. What is 2R5 rule? What is 2R5 rule? So you will say, okay, ma'am, I got it. So whenever any person in my contact having chickenpox is there, I will leave and run away. I will run away. I will not come in contact with that person. Otherwise, I will also have, as you have told us, that rate of infectivity is 90%. So I will run away, right? But when you will come to know that the person is having chickenpox, when he is having rash, at that time you will come to know, now, oh my God, he's having chickenpox, he's having rash, but he is infected 2 days before the rash. So the rule is 2R5. 2 days before the rash and 5 days after the rash. So during this period, the person is infected. So before the rash, he has already infected you. So there is no point of running now. You are in contact till now, no? So on the day of rash, before 2 days, he is infected. So like in COVID, sometimes asymptomatic person is also spreading it, no? He is not knowing that I am having COVID and he is spreading to others. The ones he diagnosed as COVID, he is having symptoms, but before that he has spread it to many one. So that is the like that only. So it is 2R5. What is the rule? So 2 days before rash and 5 days after rash, the person is infected, right? That is a secondary attack rate is 90%. As I have told you, 90% chances that the other persons in contact will be have the infection. So that is the thing, right? The usual age it occurs in children, 5 to 9 years के बच्चों में होगा. You have to differentiate the rash from the smallpox, right? So this is the rash. You can see just a second. Yeah, so this is chickenpox. You can see typical rash of the chickenpox. Can you see the diagram? I guess you can see the diagram. Yes. So this is a typical rash of the chickenpox. Can you see? It is occurring in children. This is a child. This is a child. 5 to 9 year के child में होता है. Right. Now see, smallpox का rash भी तो ऐसा ही था. But you have to differentiate the rash from the smallpox. So there are various stages. First a papule is formed, then macule is formed, then pustule is formed, then vesicle is formed, then scar is formed. So these are the various stages. Now see the zoom version. See the zoom version. Some of them are papules, some are pustules, some are pustules, some are scarring. So I have told you there is pleomorphism. What do you mean by pleomorphism? Pleomorphism means various stages at the same time. At the same time, you can see the zoom version. Some of them are papules, some of them are pustules, some of them are macule. But if you see smallpox, all are in same stage. Either all are papule, they all mature, become macule, they all become pustule, they all become vesicle, they all become scab in smallpox. But here in chickenpox, there is pleomorphism. You can see on day 2, this is the feature. On day 3, this is the feature. On day 5, this is the feature. On day 10, finally scar is formed. So this is scar. So this is papule, pustule, vesicle, likewise. Give me a thumbs up. Give me a thumbs up, everyone. So you can see first a red spot is there, then a blister is formed, and in the end scar is formed. The hard crust is formed, and all three can occur together. Some are red spots, some are blisters, and some are hard crust at the same time. Give me a thumbs up. Everyone give me a thumbs up. Everyone give me a thumbs up. So in chickenpox, palms and soles are spared. Chickenpox never occurs on palms and soles. On palms and soles, the vesicle never occurs. On axilla, it occurs. Can you see? I have shown the axilla of a child. And if you zoom out, you can see the lesions inside it. Axilla have the lesions. But if you see smallpox, smallpox never occur in axilla. Smallpox can occur in palm and sole, but it never occurs in axilla. Give me a thumbs up. Give me a thumbs up. So you can see the various diagrams. Everyone give me a thumbs up. So you have to differentiate the rash of chickenpox and smallpox. Let me teach you. I will come on the disease again. I will teach you the differences between them. Pathogenesis. So same pathogenesis. So first the virus. Both viruses are entering together. Varicella, Zoster. They always occur together. They enter via respiratory route. From the respiratory route, they will go in the trachea. Just suppose I am inhaling the virus. The virus is present in the air in the form. You have the chickenpox. You have sneezed. You have coughed. So you have thrown the virus in the air. So air have the aerosols. Inside aerosols, virus is present. And I am inhaling it. So I am inhaling the aerosol. So virus is going inside me. Virus is going inside my trachea, inside my lungs. So from my lungs, it is going in the lymph node. In the lymph node, it is replicating and coming in the blood. So it is primary viremia. From primary viremia, virus again go in the reticuloendothelial system, RES, in the liver, in the spleen. Again, it will replicate. All viruses again go in reticuloendothelial system and replicate, and then they come in the blood, secondary viremia. And after that, it goes to the skin and produce rash. It produces rash, and this rash is chickenpox. This rash is chickenpox, right? After chickenpox, it is going in dorsal root ganglion and remain latent there for years, for years, for years. After that, due to the drop in immunity, it again become reactivated. So you can see varicella zoster virus first causing chickenpox, and they remain latent in sensory root ganglion. And again, after that, it become activated and causes rash. Again, it is known as shingles. It is known as shingles. Give me a thumbs up. Whenever any person have chickenpox, there are two stages in chickenpox. First, the person have fever, back pain, malaise, shivering, cough, cold. That is flu-like symptom. Flu-like symptom. This is pre-eruptive stage. Abhi eruption nahi hua hai. Rash is not there. The person is having cough, cold, fever. I'm not feeling well. I'm having fever. I'm having headache. I'm having severe malaise. मेरे हाथ पैर बहुत दुख रहे हैं. मुझे सर्दी खांसी गला दुख रहा है. So that occurs for 2-3 days. And immediately after that, eruptive stage, the person have rash all over the body. The person have rash all over the body. There is a characteristic rash. If you rupture any of the, can you see this is a vesicle containing a fluid? You can see this is a vesicle containing. If you rupture it from inside, there is only one compartment, unilocular. It is unilocular, and the rash is very superficial. It is only on epidermis. It is not deep on dermis. It is superficial rash. So both these features are in contrast with smallpox. In smallpox, if you rupture, it is It is multilocular, multi-compartment, and the rash is deep. So here in chickenpox, the rash is superficial and unilocular. In contrast to smallpox, the rash is deep and multilocular. Give me a thumbs up if you got this point. On every point, please give me a thumbs up. Please give me a thumbs up, right? The rash in chickenpox usually occurs on the flexure surface. The rash in smallpox, in contrast, smallpox, the rash usually occurs on extensor surface. Usually occurs on extensor, but here the rash usually occurs on flexure surface. Here the rash is centripetal. In smallpox, it is centrifugal. Can you understand the difference between centripetal and centrifugal? I guess you can understand. I guess you can understand. So let me draw centripetal and centrifugal, right? So this is the mouth. Mouth is the center if I am considering. So rash if starting from mouth and going everywhere else. Starting from mouth and then going everywhere else. This is away from the center and starting from the trunk and coming towards the mouth. Starting from trunk and periphery and coming towards the mouth. This is towards central. So this is centripetal. So this is centrifugal and this is centripetal. So you tell me which distribution. Centrifugal takes place in smallpox and centripetal takes place in chickenpox. CP for CP. Centripetal for chickenpox. Give me a thumbs up. So in chickenpox, you tell me the rash is centripetal. CP for CP. Chickenpox, centripetal. As contrast, the rash is centrifugal in smallpox. Give me a thumbs up. I cannot simplify more than this. Give me a thumbs up, everyone. Right. Now, the various parts. So, axilla and palm soles. Here in chickenpox, axilla में rash होगा, but palm soles में नहीं होगा. In contrast to smallpox, वहाँ पे palm soles में नहीं होगा, but axilla में होगा. So that is the areas we can. Pleomorphic. Here rash is pleomorphic. So all the stages are together. The papule, pustule, right, vesicle, all stages are together. In compared to smallpox, rash is non-pleomorphic or monomorphic. All are in one stage only, right? Here in rash, can you see? Let me zoom it out this diagram. You can see with every vesicle, please try to see the adjacent skin. The adjacent skin around every lesion is red. That is, inflammation is there. In the background, there is the red. So in the background, there is inflammation around every lesion, right? So there is inflammation. So it is looking, the inflammation is red color. So it is looking like a rose petal, and the lesion is looking like a dew drop. So this appearance is known as dew drop on a dew, यानी समझते हैं ना, ओस की बूंदें गुलाब के फूल की पंखुड़ी जैसा. So this is a description given in the books. So this is known as dew drop on a rose petal appearance. This appearance is occurring in chickenpox, not in smallpox, because in smallpox vesicles are there, but around the vesicle there is no inflammation. So redness is not there. Redness नहीं है तो rose petal नहीं है, only dew drop है. So in smallpox there is no rose petal arrangement. Give me a thumbs up. So यहाँ पे inflammation है. In smallpox there is no inflammation, and here the resolution is very rapid within 7 days. In smallpox, resolution is late after 14 days. Give me a thumbs up. This table is going to be ultra important for you. We have covered this table in smallpox also, and we have covered this table in chickenpox also. Chickenpox is a disease which is caused by one of the herpes virus, basically herpes type 3 virus. The name of the virus is Varicella zoster virus. उसमें से भी Varicella is causing chickenpox. Smallpox is caused by a virus which belongs to Pox viridae family. Pox viridae में there are 6-7 viruses, many viruses. The most important which causes smallpox is Variola virus. Now see, the family is different, the member is different. So chickenpox is caused by herpes family virus, herpes type 3, Varicella zoster, and smallpox is caused by Pox viridae family. The name of the virus is Variola, and you know the differences between them. Now on earth, we don't have a single case of smallpox, but we have many cases of chickenpox. So chickenpox is not yet eradicated, but smallpox is already eradicated on 8th May 1980. 8th May 1980, the world is declared free of smallpox. Give me a thumbs up. I already done that. Everyone give me a thumbs up. Let me teach you the complications of chickenpox. Complication क्या है chickenpox के? What is the most common complication in children? What is the most common complication in adults? In children, whenever the children have this smallpox now, so they have itching in the lesion. So he's a child. Can you see? He will itch everywhere. वो उसको भी खुजली करेगा, उसको itching होगा हर तरफ कि ये मुझे क्या हो गया है. So he will itch everywhere. During itching, it will cause the rupture. Of all vesicles. So vesicles will get infected secondarily by bacteria. So secondary bacterial infection, and in सब के अंदर pus बन जाएगा. All of them will have pus inside them because they got ruptured and they got secondarily infected by staphylococcus and streptococcus bacteria. राइट. सो वायरस तो था ही था, साथ में बैक्टीरिया भी आ गया. सो दैट इज द मोस्ट कॉमन कॉम्प्लिकेशन इन चिल्ड्रन बिकॉज ऑफ इचिंग. Secondary bacterial infection of the skin, that is staphylococcus and streptococcal infection. Give me a thumbs up, give me a thumbs up. That is the complication in the skin. So children में most common complication occurs in skin complication, that is secondarily bacterial infection of the skin. Can you tell me most common extra skin complication, extra cutaneous complication in children? So involvement of brain. In children, brain got involved. In the brain, encephalitis can occur, meningitis can occur. But in adults, most common complication is pneumonia. Most common complication is pneumonia, and late complication is shingles. Everyone knows shingles is basically the secondary rash that occurs due to the reactivation of virus. So shingles to ho hi sakta hai. So whenever I was a kid, I guess I was 10, 12 years old, I was having chickenpox. I can remember that I'm having a deep memory. I was having severe pain, multiple lesions, and my siblings, my brother and sister, both of them got by me. I was the primary case, and I got infected. My whole home and everyone got infected by me. So in the all the children in my community, my friends, everyone got chickenpox. I was the index case, so I can remember that incident very well. And till now, I am not having shingles. But anytime I can have shingles. So virus can be present inside the body in latent, and during the drop of the, I'm sorry, at the time of the drop of the immunity, it can get reactivated. Give me a thumbs up. Come on, give me a thumbs up. You got it? So shingles can occur. So, and the complication in adults is pneumonia. So what is the summary? Tell me the complications. Tell me the complications in children and tell me the complication in adult of varicella zoster virus. I'm talking about varicella, not zoster. Who will tell me? In children, the complication is secondary infection. Secondary infection. In children, the complication is secondary infection caused by bacteria due to itching, right? Staphylococcus streptococcus. Extracutaneous complication is involvement of CNS. In adults, the most common complication is pneumonia. Pneumonia. That's it. Please learn in sab pe question hai. So that is the thing. So I have already taught you the chickenpox, right? I'm sorry. Oh my God. I was having a special class at 10 o'clock. In the flow of teaching, I just forgot it and I'm already 10 minutes late for my special class. I'm really sorry. I have to immediately stop this class. There is a notification. Okay, I'm sorry. So I have to stop this class. I will continue the lecture on the special class. Please everyone join me on the special class. Right now, I'm joining the special class on Unacademy app. Thank you very much for being with me for this class. And please everyone join me on the special class. You already know about the subscription, so I'm not telling you right now in this class for the subscription. I really forgot to take it. So I'm ending this lecture. Please everyone join me on Unacademy Learners app for the free class where I will continue this lecture. You can see me live after 1 minute there. Bye-bye.